Thank you so much for joining us today. My name is Sarah Warren. I'm the Senior Director of Translational Science here at NanoString Technologies, and I'm pleased to be talking with you today about some recent work that we've been doing at NanoString. And specifically, I'm going to be focusing on two papers that we've just been publishing that use our GeoMx platform to understand the role of B cells in response to immunotherapy. And I'm very pleased to be with you today. I'm an immunologist by training, and so I'm very interested in understanding how cellular immune responses are shaping the tumor microenvironment and the progression of cancer. And specifically, it has become obvious in the field recently that there are structures called tertiary lymphoid organs that arise within tumors that are very important for shaping that immune response. So tertiary lymphoid structures are ectopic lymphoid organs, which means that they arise in places where there's not normally lymph tissue, and they occur in places where there is chronic inflammation ongoing. And what it is that it is an immune organ that actually forms and organizes itself into regions of T cells and B cells and follicular dendritic cells that all come together and interact and promote immune response. And then that immune response can help to propagate and shape an immune response within the tumor microenvironment and directed against tumor antigens. So specifically within these tertiary lymphoid structures, as you can see on the slide here, there are many different cell types that are involved. They are very structurally organized, and the consequences of activity within the tertiary lymphoid structures, or TLSs, is that we see increased levels of T cell differentiation and T cell maturation into effector memory and effector cells, so cytotoxic T cells, memory B cells, plasma cells that are generating antibodies, and these can then impact those immune responses against the tumor. So there's been a lot of investigation over the past few years about really what is the impact of these tertiary lymphoid structures on the prognosis of tumors. And many studies have been done to understand if they have a prognostic effect. And the results have been mixed, but generally suggesting that yes, having tertiary lymphoid structures is positive for disease prognosis, disease outcome, good outcomes for these patients. I'm showing here just 3 examples from colorectal cancer, non-small cell lung cancer, and pancreatic cancer that all suggest that having tertiary lymphoid structures increases the rate of disease-free survival. But the role of TLSs in melanoma is less clear, right? There have been mixed reports about their clinical impact, and at a molecular and cellular level, understanding what effects they're having on the immune response to melanoma are studies that had not been done previously. So recently, 2 groups undertook an effort to answer some of these questions and really try to characterize the impact of the TLSs within the context of melanoma. And these papers were published in a very recent issue of the journal Nature. And you can see on the left side of the slide, one of these papers came from the University of Lund in Sweden, and the other paper came from MD Anderson Cancer Center in Houston. And so these were studies that were done independently, and each took a multi-platform approach to really dissecting the role of the TLSs in melanoma. And you can see on the right slide, I've illustrated just some of the technologies that were employed in this characterization. And the one we're going to be focusing on today is in the upper right-hand corner called DSP, Digital Spatial Profiling. What is DSP? DSP is a platform that was invented at NanoString and is now commercially available. So it is a system that we call the GeoMx Platform, and it is an instrument which enables spatially resolved molecular characterization of tissue samples using FFPE as a starting material. And what it does is it images the tissue, and it also performs molecular profiling from regions of interest within that tissue to allow you to do very high-plex analyses of the distribution of proteins and RNAs within that sample. So in a bit more detail, here is what the features of that system look like. We use a series of detection reagents, which are either oligo-labeled antibodies or RNA binding moieties, which have indexing oligos on them separated with a photocleavable linker. And that linker becomes very important when we take those detection reagents and we put them onto an FFPE slide. And then we can image the slide using immunofluorescent markers and select regions of interest that we want to profile. And these regions of interest can be as large as 600 microns squared, or it can be as small as a single lymphocyte. And once we've selected those regions of interest, we shine UV light just in that area, and that cleaves that linker, releases those oligos, and then allows us to collect and quantitate those oligos using either our nCounter platform or next-generation sequencing technology. So what we get at the end of the day is both a digital picture of the slide using 4-color immunofluorescence, and that you can see in the upper right-hand side of this slide, and also molecular data about the specific regions of interest that you have chosen to profile that allows you to do high-plex characterization of the proteins or the RNAs. And what's really fantastic about this technology is that we have a lot of flexibility over the size and the shape of these regions of interest that we're going to be profiling. And that's really useful because we know that tissue heterogeneity is really driven by underlying interactions between cells within a specific region. And so as we have tumor cells interfacing with stromal cells or immune cells in a tumor microenvironment, we want to optically dissect those two compartments, and we can do that using the GeoMx platform. And by controlling the shape of that UV light, we can separate and profile those two cell types separately. I'm not going to go into the details of the technology, but I would encourage you to come to the NanoString website to learn more about how it works, or to reach out to your NanoString representatives who would be happy to provide you with additional information. But we have built this platform, and we've also then been able to build a library of detection reagents that allow you to profile the targets that are of greatest interest for doing immuno-oncology research. So on the left side of this slide, you can see that we have a protein profiling library that is customizable because there's a central core of targets that you're going to be profiling, and then you add on modules as you need to address the biology that you are specifically interested in. And there's also the possibility of adding custom proteins at the same time. So if you have antibodies against a specific target, we can add labels to those, and then you can collect that data alongside all of the other data and antibodies that we've already validated. Today, we have over 60 antibodies that have been labeled and validated in our process. They can all be used simultaneously on a single tissue slide. And they are really focused on being able to profile holistically that immune response. When we move over to the RNA side, we have even more options here because now we don't have to rely on having available antibodies, which, as you know, is not always an easy thing to develop. But here we have a panel that's called our Cancer Transcriptome Atlas. And this is really contains the vast majority of genes that are necessary to characterize tumor immune interactions. It contains all of the best targets from our most popular nCounter panels, including IO360, pan-cancer pathways, pan-cancer immune, as well as additional targets that we have come to appreciate are important in regulating those tumor immune interactions. So in total, there are more than 1,800 characterized genes that are in this panel, and we can collect molecular data about all of these targets, again, with a single experiment, with a single tissue slide. And so we've seen a lot of success with this technology, and our customers and our collaborators are also having a lot of success using GeoMx to uncover fundamental, high-impact facets of immuno-oncology biology. And we've had a lot of success publishing. Here you can see some of the 13 publications that have been generated to date, appearing in very high-profile journals like Clinical Cancer Research, Nature Medicine, as well as the journal Nature. And those, I'm really going to be telling you the story today of 3 of those publications. And the first that we're going to start with was actually the first GeoMx paper that was published. And this was a paper that came from MD Anderson from the laboratory of Dr. Jennifer Wargo, and it appeared in Nature Medicine in 2018. And in this study, her group was looking to characterize the immune response that was elicited by neoadjuvant and adjuvant immunotherapy in patients with stage 3 melanoma. And she was again taking a multi-platform approach to characterize all of the immunological changes that were induced in response to treatment, and also to understand which of those were associated with favorable outcomes. And what she observed from that paper, from doing the GeoMx profiling here, was that there was an increased abundance of proteins that were associated with B cells that were observed in patients that had favorable outcomes. So you can see, using 2 different antibodies from a single experiment, we got independent validations that both CD19 and CD20, which are key molecules expressed exclusively on B cells, were expressed at higher levels in patients with good response. And this was exciting and intriguing results because, as I mentioned before, the role of B cells in regulating tumor immune interactions has not been fully elucidated. So she wanted to go on and dissect this role of the B cells in greater detail. And to do that, she turned to a different clinical trial that she had samples available from, where again we were looking at patients with advanced melanoma, and this time giving them immunotherapy as a neoadjuvant prior to surgery. We're very fortunate that this is the result that was published in January of this year in the Journal of Nature. And so what she was doing with the GeoMx profiling specifically was to look at FFPE tissue sections from the melanoma patients, from the pre-treatment biopsies, and focusing specifically on the tertiary lymphoid structures. And so in this slide, you can see that I've highlighted the slide images from just one patient, and we've got the immunofluorescent images that were collected on GeoMx on the left, and then the corresponding H&E image on the right, and we've highlighted the TLSs for you here. And within those TLSs, we were then dissecting out optically using that UV light to characterize T cells and B cells separately. And what we observed here was that in the T cell compartment, for T cells that were in the TLSs, they had higher expression of the activation markers and of checkpoint molecules than did T cells that were located externally to TLSs. So this is suggesting that, as I described to you at the beginning, that actually, yes, the presence of T cells in these TLSs is promoting their maturation, is promoting their differentiation. And the graph that we're looking at on the left here is showing you the aggregate results from each of the individual patients that we profiled. And on the right side of the slide, you can see for each of the individual patients, how their expression of these particular markers was varying. And so there's quite a heterogeneity between the patients and some overall trends for the most robust genes that were observed across all the patients. And the other thing that we've done that's not represented on this slide, but is certainly something that is possible with the GeoMx profiling, is to then look within each of the patient's individual samples and look at the distribution of protein expression within the different TLSs, or within those different regions of interest that we profile, to add another layer of complexity and variability in the data, and even more finely dissect the molecular expression patterning that is controlling those immune responses. And when she looked in greater detail here at the level of TLSs, at the level of the B cells specifically, she was able to identify that B cells were associated with proliferation, as indicated by Ki67, and that we were seeing in some patients differences in that level of B cell activation in the TLSs, which also suggests then that not only are the T cells becoming activated, but the B cells themselves are becoming activated, developing into memory B cells, developing into potentially those plasma B cells that could be secreting anti-tumor antibodies to further mediate the tumor immune responses. And the results that she generated on GeoMx were also supported by other profiling characterization that she did on the samples. So I'm not going to go into great detail, but again, I just wanted to show you very briefly that the RNA-seq data that she generated also suggested that there were higher expression of B-cell associated genes in the TLSs, as well as immunofluorescence and immunohistochemistry staining that corroborated the role of TLSs and B cells and T cell activation happening here in the context of the TLS in the tumor microenvironment. The second paper then that was published in the same issue of Nature, as back-to-back article, is a paper that came from Lund University from the laboratory of Professor Goran Johnson. And Dr. Johnson had available to him tissue microarrays of biopsies from melanoma patients, and some of those biopsies contained tertiary lymphoid structures. So we wanted to use the GeoMx platform to characterize the TLSs here and the non-TLS tissue as well, so that we could understand whether the same trends that were observed in Dr. Wargo's studies could also be seen in these studies. And so the profiling strategy here was similar. We had two different TMAs to profile. A fraction of them had TLSs in them. Here we used a masking strategy that allowed us to optically dissect out the protein expression patterns from T cells, from the tumor cells, and from B cells separately. And so you can see the masking that we did here based on each of those ROI selection strategies. And that allows us to, again, very finely discriminate where the protein signaling is coming from. And this is not a level of dissection that's really available on any other profiling platform. So Dr. Johnson observed when he looked at the B cell signatures that there were two different populations of B cells that were present within these tissue samples. And they were really separated based on their level of Ki67. And so this suggested to him that there are TLSs there at different states of maturation, where as the TLSs form, the B cells are more quiescent, and then as they go on to develop and mature and form more of a architectural structure, the B cells become activated and then are potentially able to have more impact on regulating the T cell activation. And his data that supported the hypotheses around B cells regulating T cell activation is what's shown on the right side of the graph here, where when he looked at the T cell areas of interest that were from T cells that were close to or within the TLSs versus those that were very far away from the TLSs, he observed that there was more expression of CD4 and more expression of the anti-apoptotic protein BCL2 when the T cells were located within those TLSs. So this is suggesting that they've become activated, that they are differentiating, that they are seeing more exposure to the antigen, and may be becoming more activated to mediate a stronger anti-tumor immune response. He also was able to look at the level of the T cells, specifically in greater detail here. So you're seeing now not just those those 3 targets, but we're looking here also at TLSs from T cells from tumors that had no TLS present within them, T cells from tumors with TLS, and T cells from tumors out within the TLS themselves. And we're seeing here again different molecular patterns, different patterns of expression of these targets that is suggesting that these T cells are existing within different states. And you can see that indicated more clearly for the individual cores that were on the TMA in the box and whisker plots that are on the right. So those T cells that are inside the TLSs tend to have different patterns of expression of the activation markers and the immune checkpoint markers themselves. And then he went on as well to characterize the melanocytes and the actual tumor cells that could be regulating also the activation and activity of the T cells once they'd left the TLSs. And here, what you can see on the left is that he's looking at, again, the different molecular patterns that are expressed by the melanocytes from tumors with or without TLSs or these lymphoid aggregates. And what you can see is that there are very different patterns. There are the canonical cold tumors in the middle indicated in the yellow bucket where there's very little expression of activation molecules or antigen expression molecules by the tumor cells themselves. He then was able to look at B2M copy number and see that there was increased presence of loss of B2M in that particular sample population compared to looking at tumors that had T cells or tumors that had the TLSs. So this is really suggesting that there is biology that is being regulated by the TLS and that is promoting anti-tumor immune response. And it's also indicating that all of this was data that was collected on the GeoMx platform. So this is a platform that is very useful for dissecting out those underlying biologies and revealing a great deal of the complexity of tumors in a way that is requiring a very little amount of tissue and is very tissue sparing. So all of the data that I have shown you here was collected from a single slide from each patient, and so it's really a very large amount of data that is being generated from a very small amount of clinical tissue, which is very helpful when you're often working with small amounts of patient tissue and it's difficult to characterize any molecular signals in great detail. So I hope that I have shown you today that the GeoMx platform can be used for spatially resolving the RNA and protein profiles from the tumor microenvironment, and that it can be used then to reveal biology that would be otherwise invisible if bulk profiling is all that's performed on the tissue. I hope you've also seen that GeoMx experiments can translate very well across labs. So there is very little user-to-user variability, site-to-site variability, and so it generates very highly concordant results if the underlying biology that's measured is similar, which really enables cross-lab collaboration, comparison of different data sets to quickly drill down in on that biology, which is ground truth, which is most impactful then for regulating tumor immune responses. And I hope you've also come to appreciate that incorporating spatial profiling into your translational research has the potential to lead to the development of more predictive biomarkers that may someday be able to be used to improve patient outcomes to immunotherapy by allowing us to target those patients which are going to be most sensitive to any given IO therapeutic. And our hope is that the GeoMx platform, and in combination with profiling on other platforms, such as our nCounter for bulk tissue profiling, leads to a cycle of continuous discovery that is going to accelerate the pace at which we are able to learn about the underlying biology of tumor-immune interactions, identify new drug candidates, develop and test those drug candidates very quickly, and then develop the biomarkers associated with them that will allow us to give those therapies to the appropriate patient populations. So I thank you for your time in listening with us today. I would encourage you to visit our website of GeoMx at nanostring.com to learn more information about the GeoMx Spatial Profiler, and we'd be happy to answer any questions that are submitted through the email alias that's going to be presented in conjunction with this webinar. Thank you very much. Have a great day. Goodbye.